I'll start. I'll start since no one has joined. I will just go through assignments, week 4 assignments by myself, and I hope whoever is watching later on YouTube, they will find it helpful. So the question number 1 is photosynthesis. Photosynthesis is a proof of which law in operation in biophysical system. So this one will be photosynthesis. What happens is it converts a certain kind of energy to another kind of energy. So this first law of thermodynamics says that energy cannot be created or destroyed like in normal terms, if you want to say, not in mathematical terms. It can be converted from one form of energy to another form. So that's what happens in photosynthesis. So that is the first answer. So what happens in photosynthesis is heat, light, which contains photons, that contains energy. This gets converted to chemical energy and stored in leaves. So this is first law of thermodynamics, which says that energy will be converted from one form of energy to another form of energy. So question number 2 is bacterial cells, considering the model E. coli, have a shape of spherocylinder of length 2 micrometer and diameter 1 micrometer. What is the closest value of this volume and mass of such an idealized bacterium? Choose an answer in the order of magnitude. So what is a spherocylinder? So spherocylinder looks like this. It's called spherocylinder slash capsule. So first we need to find out what's the volume of it. So if you can see here, it actually consists of one sphere and one cylinder, and this is an hemisphere, this is another hemisphere. So the volume will be volume of this sphere plus half volume of one sphere, which is like this, plus half volume of another sphere, which is this, plus volume of cylinder. So this makes volume of sphere plus one cylinder. So one sphere plus one cylinder, which this is 4/3 pi. The radius of the cylinder is r cube plus pi r square times a. This is the volume. Now the question is volume and mass of such an idealized bacterium. So you know here a is known and r is known. And how do you calculate mass? What is the mass? So mass is coming from the density. So in E. coli, it's mostly full of water. So you use 1 gram per cc and calculate this mass. So m is okay, so you put it all back. So this is how you calculate volume, and this is how you calculate. So don't forget to convert this gram per cc to proper unit, or these micrometers to proper units, and you will get some value. And here it is given in liters, so you have to convert it to liter also. So that's how you can calculate the volume and mass of E. coli bacteria. So we'll go to the next question. How many proteins are there on the order of magnitude in an E. coli cell? So you can find out. It will be like around 10 to the power. So how many proteins are there in the there in order of asking only on order of magnitude, so it's around 3 to 4 million, which is 3 to 4 times 10 to the power 6. So it will be around 10 to the power 6 order of magnitude in E. coli. So what is the genome length of the human cell diploid if the DNA were stretched n to n with all chromosomes lined up. So use the approximate to the diameter to represent the size scale. So if you need two information, what is the total length of human genome? It is 3 into 10 to the power 9 base pair. So what is this base pair? Base pairs are this ATGC bonds. So this is said in the units of base pair. Now so each base pair, now we have to answer it in terms of meter, millimeter in those units. So one base pair is 3.4 angstrom. Now so the total will be 3 into 10 to the power 9 times 3.4 10 to the power minus 10 meter. So if you calculate this, you will get the right answer. So what is the scale of the green fluorescent protein GFP? Choose the order of magnitude closest to the correct answer and the approximate diameter represents size scale order of magnitude discussed. So it's around nanometers scale, and for reference this is the GFP. GFP is very interesting protein which like it's a green, its color is green and it's used to detect cells, nucleus, everything like in real experiments. So this is a good nice information to have. This is green fluorescent protein and this is the diameter. What is the osmotic pressure gradient acting along the moderate E. coli sustainable pure? So this question is I have already discussed in a previous tutorial, which the formula for osmotic pressure is ICRT. C is the concentration of the and I is the this is for one for non-electrolyte. So this is one of factor as I can remember. R is the gas constant, T is the temperature. So if you put all of this, you can get the correct answer. questions: which of the following drive diffusion of molecular solutions in the absence of concentration gradient? So this is a very nice question. This question also has been discussed in a previous tutorial, so I'll still give you some idea about it. So diffusion of molecules involves particle Brownian motion of particles. Let's say you have in a box multiple particles like this. So it is at a finite temperature T. So if it has a finite temperature, particles have kinetic energy. This means particles have velocity. So kinetic energy is directly related to velocity, we all know. So if particles have velocity, so they will move around like this. Everyone will move around in some direction. But there are other particles also. So what happens is, if a particle is trying to go in a particular direction, it will find some other particle there, and it will get reflected by this. So and there will be because of the crowded environment, particle cannot go in a particular sustained direction for longer time. It keeps moving in zigzag pattern. So this motion is called Brownian motion. So this Brownian motion of particles lead to diffusion. Diffusion even without absence of gradients. So the correct answer will be thermal energy. So it is done. This also has been discussed. So what it's actually asking is asexual dividing cells going by binary fission. So what binary fission is let's say you have one cell or one bacteria, then it becomes two. So this again divides to two more. So this is called binary fission. Okay, so if you see, like at n t, if n is the number of cell at a time t, and n0, which is equals n at t equal to 0, that is number of cells at time t equal to 0. Now let's say t equals 1, n1 will become 2, t equals 2, n2 will becomes 4, right? So t equals 3, it becomes 8. So nt is actually 2 to the power t. So I should write so you can follow the pattern, right? So nt becomes 2 to the power t if n0 equals 1 at t equal to 0, 1. So if you have a finite n0, so you will find nt is n0 2 to the power t. Now there is this concept of doubling time. So doubling time means at what time nt becomes twice of n0. So that t equal to tt, that time will be tt. So if you put ntd equals n0 2 to the power td. So 2n0 td. So td in this case becomes 1. If you don't have so it is already assumed that td equals 1. That's how you get this. If you have a finite value of td, that will become 2 to the power t by td. So no n0. So this is the answer. So minimally how many membrane layers already done. Fibroblast, it is 2. In the cells it has 2 layers. So when going in it will have one face one bilayer, while coming out it will face another bilayer. What percent of the space of human fibroblast nucleus occupied by genomics? It's will be 0.2. You can find it anywhere. So this will conclude the discussion about week 4 assignments. So these are all week 4 assignments. So next I will discuss the some of key points that have been in the lectures to motivate you to actually watch those lecture series. So this is the topic is how to estimate tile scales. So please go through the usual video first. These have been discussed there in details. I would just give touch on few topics. I will not go into the details. one important and very interesting topic he has discussed in the main lectures. That is how to estimate time scales. So what is means by time scale? So every event occurring in this universe, in this nature, in any scale, any length scale, you will have some kind of times attached to it. So, so to define the size of anything in this universe, we use length scale. So for microorganisms and everything, the length scales are at micrometer order, let's say in this order, or in uh, celestial planets they are in the like light year order, so if I say, okay, the length scale is in micrometer or meter or in light year or in billion meters, so you know how big things are, and what how further one thing is from another. Same thing with time scale. Time scale tells you how fast stuff are going on, how fast something is moving. Like let's say for for planets and stars, like you can express their age with associate it with some time of a of a star. The speed of a planet you can associate a time to that. Similarly, in micro scale you have these cells, so cells can move around. Bacterias, they also can move around. So to have a time scale, or let's say cells are dividing, so from here cells are dividing, so how long does this whole process takes? So like from one cell to divide, so you can convert into seconds and have an idea with your regular life, like when you are doing something, from you walking from here to another place, let's say it took like a minute, and how many cells have been divided in that minute. Those kind of ideas you can have by having ideas about time scales. So in biological systems, in experiments, these time scales have uh, the way to measure time scales are mostly of this three kind. One is fixed time point analysis, one is direct observation, one is pulses. I will just briefly give you idea about these two: fixed time point analysis and direct observation. So what is fixed time point analysis? Fixed time point analysis, if when you look at a whole process, not continuously but of a discrete manner, like you take snapshots of different stages of that whole event and have and try to take some idea about that event. Let's say this fruit fly Drosophila, so it's from pupa to become adult, then again giving embryo, and eventually becomes pupa. And this whole life cycle, you don't need or you don't need or you don't have to look at every single step of this process. What you can do, you can just take snapshots of some significant changes. Like let's say you have embryo, then from embryo after one day it becomes first insta larva, then it becomes second insta larva, like this. Eventually every one day you take snapshot and you get another form of this species, and you see it is growing. Now everything is happening in one day. Now you see, so after 2.5 to 3 days it becomes pupa. So this is not same as this one day interval. So you see a significant change after 3 days, it's pupa. Then you see this 3.5 days after becoming pupa, it becomes an adult, and it's either male or female. Now you can say the total time from embryo to becoming adults, you can estimate from there. It's around 6, like around 10 days. In 10 days, from a embryo to a food fly fully adult food fly, it takes a time. So this is how you get an idea of the time scale of these flies. Also there is this bacterial growth. So bacterial, currently when it grows, it has different phases. So this also you can have via fixed time point analysis. At different time you take different snapshots. So you there are like different stages of this growth. First it is lag phase, where there is not significant growth is observed. Then it starts growing exponentially. It's very fast growth. This is lag phase. Then it starts have this exponential growth. Then eventually it saturates. To some point, and in future it dies death. So you can associate an equation to it, which is, it looks like this. This is this is like if you analyze this equation, nt equals n0. So let me sorry. So if you see this nt, so this r is the rate, and this is the saturation. This k is the saturation value, and n0 is the initial value. So if you put t equal to 0, so you see n0 becomes n0 k. So nt becomes n0 k by n0 plus k minus n0. So this gets cancelled out, k gets cancelled, so it becomes n0. So n0 is the initial. Now if you look at t tends to infinite, so nt tends to infinity, it's n0 k by, so this whole term is now 0, because if t tends to infinity, e to the power minus rt goes to 0. So by n0, so n of t tends to infinity is k. So in this graph, this point is n0 and this is k. So in this equation does not associate you to this. So this is how you can get a time scale of how bacterial growth happens. So this is fixed point time analysis. Next is direct observation. So in some instance you can have direct observation via direct experimental observation. So let's say you had this one bacteria and you keep observing it, and you get to this kind of colony formation. So this was one of the results from Athalasar's lab. So this kind of treatment is also necessary to have continuous information of how things are changing. And the last one is pulse chase. I encourage you to actually watch the videos and have some ideas about it. So last topic I will discuss is bacterial motility, or some in general cell motility. So what is motility and what kind of behavior a bacteria shows in real environment, and from there I'll give some hints to calculate actual speed of a bacterial motion. last topic we are going to discuss today is how the bacterial motility. So first is they show chemotaxis behavior. So what is chemotaxis? Chemotaxis means taxis. Chemotaxis in form is it means that when bacteria follow in some kind of chemical concentration gradient. So let's say there is some food here. So if it in a biological system, this food would not stay here. It will have some gradient, like it will highly concentrated here, then low concentration. Eventually it will have some kind of effect to at a particular distance. Now if a bacteria is here, so it can feel this chemical gradient and it can climb up the gradient to reach the center. So this is called chemotaxis. Then there is random walk. So what happens in random walk is some bacteria do a run and tumble motion. So run and tumble. So this chemotaxis does not happen like a straight line motion. So if they go in a straight line for a particular amount of time, then they change its direction due to its internal mechanism, and that's how they find this. Let's say it was going in this direction and it sees this chemical gradient, it can rotate itself towards this and go like this. So this is another example. So this is a run and tumble motion. And last one is flagellar rotation. So what is flagellar rotation? So it has this flagella associated with this, like this is the flagella part, so it flaps it. It's like when you are doing boating, it does when you move the paddle around, it puts force to that medium, to the water, and help you move. Same way it flips its flagella in a periodic manner, and it helps the viscosity of the medium helps it go forward. So this kind of motions bacteria does in a particular medium usually. So from all of this information you can calculate what will be the speed of a bacteria moving in a biological system. So to calculate that, I encourage you to go through the lectures and you will have informations that will help you calculate this bacterial motion, and you can have an estimate of is a bacteria is faster than a human or not. So that's how you get an idea about time scales. Again, this concept of time scales comes back, so you can have the how fast things are moving, as we have already discussed. So that will end today's tutorial. Since no one has joined, I am not going to discuss anything with anyone. So okay, thank you.